Hi, I'm Kaitlyn Okin, and I am a junior at Hollins University studying biochemistry and public health.
Hi, I'm Alea Rodriguez. I'm a sophomore undergraduate student at Hollins University, and today we're going to be talking about cysteine containing proteins and Maleimide probes. So let's go fishing. To begin, we will go over our research aims for this project. Our first aim is to design the Maleimide probes via synthesis and evaluate the specificity of these probes against cysteine-containing peptides. Our second aim is to determine the various activities of cysteine-containing proteins using our design probes, such as determining changes in oxidative states and the activity within patient cytoplasmic and serum samples. So most of us understand that amino acids are the monomers that are used to build proteins, and that proteins play a huge role in metabolism within the body. So it is very important that there are balanced concentrations of amino acids within our bodies in order to regulate biological processes. Too much or too little can result in various health-related issues. In the case of the concentration of cysteine, when there are too much, one can suffer from neurotoxicity, cardiovascular disease, and Alzheimer's. While on the other hand, with too little, one can experience hair depigmentation, lethargy, and overall weakness. So just from the fluctuations of cysteine alone, there can be major impacts within the body that could potentially be better regulated with our probe design.
Cysteine is important for maintaining redox homeostasis, regulating cell signaling, and overall protein structure. Cysteine in metabolism can sometimes mutate and is no longer cysteine. These are examples of how cysteine has been mutated. The mutation of cysteine changes the function of the overall protein, and that can either be favored or not depending on the change that is done. The thyrodoxine system is a very important system in synthesis. It is essential for normal cellular function, redox control of transcription factors, synthesis of deoxyribonucleotides, cell growth, and protection against oxidative stress. In this example, you can see the reduced thyrodoxine catalyzing the reduction of disulfide bonds within oxidized cellular proteins. One of these oxidized cellular proteins is peroxyredoxin. This is an example of how the disulfide bonds contribute to the folding of proteins. It is especially important in the tertiary of a structure where the actual folding occurs. One way to think of this application is as curling your hair from straight hair. The reactions of between the different proteins go through a reduction phase, which is then heated up and causes the protein to start to curl. After it starts curling, it is then oxidized and folds into its proper structure, much like when your straight hair becomes curly.
There are various available probes that are currently in use today that can be applied with analytical methods and fluorescence and are used for intracellular detection. There is the Michael addition style, cyclization, substitution reaction, cis-metal binding, and disulfide bond cleavage reaction that are all detection mechanisms that the probes are based on. The use of fluorescence is produced either of two ways, either from the attack of thiol from dye reagents or the cleavage of fluorescent moiety. One of the most common fluorescent moiety is rhodamine dye due to its wide range of emission. Although these all work wonderfully, there are disadvantages that we aim to overcome with our specific probe. One disadvantage to using these probes may be the transition metal ions associated with them provide little spectral changes, thus making them difficult to detect and provide unwanted background results. A major disadvantage that we are also looking at is the toxicity of such metal ions in the environment and our health. For example, mercury is known to easily cross biological membranes, and even with the smallest amount can lead to digestive, kidney, and neurological diseases. Nonspecific binding of these metals can also decrease the sensitivity for the detection of our protein of interest. And so once more, with our probe, we aim to overcome such disadvantages that are currently seen with these current models. Yes. Before we dive into further detail of the mechanism of our probe, we would like to highlight some of our research goals. So first, we would like to gain a better understanding of the role of cysteine-containing protein activities, specifically in cancer and degenerative diseases associated with aging. And we would also like to gain a better understanding of the function of cysteine residues. And so in the future, we would like to apply our knowledge and our probe to help patients with cancer and other age-related diseases that suffer from the consequences of cysteine-containing protein activities.
Our current research goals are to investigate cysteine-mediated protein activities using chemical platforms. We will synthesize these probes through synthetic chemistry, cell biology, proteomics, and mass spectrometry. Again, we would also like to identify the therapeutic targets and small molecule drug leads for the diagnosis and treatments of diseases such as cancer. Maleimide probes have covalent bonds formed between the thiol and alkene substituent in maleimide. To stop the synthesis of our probe, we will be doing that by using beta-mercaptan ethanol. The advantages to maleimide probes are such as identifying intact proteins without the need for upfront processing. They also act as chemical handles in order to enrich and isolate cysteine-containing proteins from complex mixtures. They are also used to analyze qualitatively and quantitatively the level of cysteine-containing proteins. This is also known as the concentration of the cysteine-containing proteins. It is also compatible with a mass spectronomic technique to confirm the covalent attachment. This is an example of the synthesis. In this example, this is how we are going to perform the synthesis of our probe along with detecting the proteins. This is the protein that will attach to the maleimide right here and go through the first step of the synthesis. When the protein attaches to the maleimide, it will attach to one of the carbons that were on the double bond. Depending on which compound we use, we will either do this with rhodamine to get a rhodamine probe or biotin to get a biotin probe. Once the binding of the probe occurs, the specific biotin or rhodamine probe will only bind to proteins that have the cysteine attachment. So as you can see here, there's a pool of proteins, and the biotin or the rhodamine are only binding to the B or D protein because those are the only two proteins that have the cysteine attachment. This is another example of synthesis that we could do if our first synthesis that I just showed doesn't work. It is a similar concept where the protein is attaching to the maleimide, but it is done through what is called click chemistry. Although click chemistry is kind of hard to explain and a little bit more complicated, so I do not have enough time to do it in this specific presentation. But if there are any questions that you have about click chemistry, I can explain them at a later time. But as you can see, the detection of proteins is similar, where only the biotin and the rhodamine will only bind to the proteins that have the cysteine attachment. In the future, we hope to be able to continue to try and detect the cysteine-containing proteins, along with identifying specific proteins and further exploring the role and activity these proteins play in related diseases.
Not only could this research benefit patients and others seeking more knowledge of the topic, but it benefits undergraduates like myself and Kaitlyn by giving us the opportunity to apply our own unique techniques and build new skill sets that can be applied to various future educational and career aspirations.
We want to thank the rest of the members of our lab, along with our mentor and the faculty in the chemistry department, along with Hollins University. Without any of these people, none of our research would be possible. If there are further questions that were not answered in this presentation about our research, feel free to reach out to us through our emails. Thank you so much for listening to our presentation today.